Okay, this is the Professor, CBMD PhD, and today's Friday, July 21st, 2023. It is 5:29 and 17 seconds, a.m. Eastern Standard Time, and today's research, brain tumors. So let's jump to it here. Efficient boron neutron capture therapy for brain tumor with boron carrier, date July 13th, 2023. Source, Tokyo Institute of Technology. A new boron agent drastically improves the effectiveness of boron neutron capture therapy for glioblastoma. The agent is selectively absorbed by brain tumor cells, exhibits enhanced blood retention, and can be administered at low dosage. Experiments on cell cultures, mice and rats, show promising results, highlighting the potential of the agent for radiotherapy. Glioblastoma is a highly aggressive form of brain tumor that originates from the supportive cells of the brain called glia. Due to its rapid growth, surgical removal of glioblastoma is often difficult, leaving radiotherapy as the most visible option. Among the many existing radiotherapeutic modalities, boron neutron capture therapy has attracted a lot of attention for the treatment of glioblastoma. As boron neutron capture therapy exploits the high affinity of boron-10, 10 sub-B atoms for low-energy neutrons. When 10 sub-B absorbs a neutron, nuclear reactions release high-energy particles that damage the nearby biological tissue. Therefore, the essential requirement in boron neutron capture therapy is to achieve a significantly higher concentration of 10B in tumor cells than that in healthy normal cells. However, this has proven to be challenging, resulting in low survival rates and limited use of boron neutron capture therapy. The scientific investigators and researchers has developed a boron agent that shows great promise for boron neutron capture therapy. This molecule, phytyrocolossal dodecacaberate, conjugated with a 4P Idaho phenylbutyric acid, moiety PBC, IP, as idophenyl dodecacaberate, idophenylbutyric acid, consists of three main functional groups. The first is a boron group, containing 12 B atoms. The second is a ligand design that binds a folate receptor. This receptor, hardly present in normal cells, is greatly overexpressed in various cancers, including glioblastoma. Thus it acts as an entry point for phytocolossal dodecacaberate, idophenylbutyric acid in tumor cells. And finally, the third group is the 4-p-idophenylbutyric acid, moiety which binds the entire molecule, albumin, an abundant carrier, protein in blood, and transport substances throughout the body. As phytocolossal dodecacaberate, idophenylbutyric acid binds non-covalently the naturally present albumin, which allows it to directly interact with tumor cells, promising its cellular uptake. Thus, the acid moiety can enhance the blood retention of the boron agent, thereby potentially reducing the required dose. The researchers conducted several experiments to confirm the viability of phytocolossal dodecacaberate, idophenylbutyric acid for boron neutron capture therapy, finding that it accumulated in glioblastoma cell cultures 10 to 20 times more than L4 boron phenylalanine, a clinically approved boron agent in Japan. In addition, phytocolossal dodecacaberate, idophenylbutyric acid showed no signs of toxicity to cells on its own, demonstrating its safety. Likewise, phytocolossal dodecacaberate, idophenylbutyric acid, administered intravenously to the human glioblastoma xenograft model, showed higher boron accumulation in tumors than boron phenylalanine, effectively suppressing tumor growth after thermal neutron irradiation. The scientific investigators and researchers say these promising results were also replicated in vivo, both in glioblastoma, xenograft models, and rat glioma models. As phytocolossal dodecacaberate, idophenylbutyric acid, administered via convection, enhanced delivery in the rat model, achieved tumor to normal brain and tumor to blood boron ratios of 37.8 and 94.6, respectively, 3 hours after completion of convection and hands delivery. And survival rates at 180 days were 50% and 70% following boron-neutron capture therapy with phytocolossal, dodecacarbonate, idophenylbutyric acid, olein, phytocolossal dodecacarbonate, idophenylbutyric acid, in combination with boron phenylalanine, respectively. There were no residual brain tumors. Overall, the proposed boron agent may represent a breakthrough in radiotherapy for glioblastoma, with the researchers currently conducting preclinical studies. And this is a professor of microbiology, bioinformatics, pathophysiology, and teacher of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.